Hi, everyone. Welcome to workshop, part of the Medical Microbiology and Infectious Disease Bioinformatics workshop. Today's workshop is going to be about Beginner's Guide to Phylogenetic Trees. I'm very much a beginner myself. My name is Taylor Davedow. I'm a PhD student in the Department of Medical Microbiology and Infectious Diseases at the University of Manitoba. So just some information for participants. Just keep in mind that all workshops are being recorded and they'll be posted to our MMID workshop YouTube. If you have any questions, you can type them into the chat, or you can go to slido.com and use today's workshop code, and they'll be read through there. This is the second last workshop in the that's going to be instructor led. Next week, we are going to be doing kind of building off from today and doing an introduction to tree visualization and annotation using ggtree based off of the tree that we built today. And then the last two workshops are kind of open to bring your own data sets and work with your colleagues and friends and analyze data together. So for anyone here in person who needs to connect to the University of Manitoba Wi-Fi, and if you have any issues, let us know. The instructions are in the slide. The objectives for today's workshop is to use a publicly available data set to build a phylogenetic tree, as well as provide a quick view of the tree output. So just kind of like a disclaimer that Jill had provided in her workshops as well, that to provide basic working instructions, all tools will be run with default settings. However, careful consideration of analysis parameters in the context of your own research questions should be taken into account when you're analyzing your own data sets, because the default parameters might not always provide the most optimal results. So we're going to get started. We'll start by opening up the terminal and navigating to your workshop directory. Okay. We're going to make a directory called Philo. So make directory and then Philo, press enter. Then we're going to list the contents of the directory to see that it's been created. And yep, it's there. For me, I also had pre-downloaded the multiple sequence alignment that we're going to use today, but we're going to do that together as well. So go to the Google Drive. Is the link in the, so go to the Google Drive and you'll be able to just click and download the msa.fasta.zip file, and then we'll unzip that together. Maybe I can get a thumbs up when everyone's gotten to that part. And once it's downloaded into your directory, make sure it's in the directory that you are currently working on. Then we can unzip it. So to unzip the file, we'll go unzip MSA, and then you can tab complete. And it should be there. And press enter. And you can see it's working. And to check if it's unzipped, you should see a second file there that doesn't have the .zip. So kind of the basic workflow, building off from the workshops of this year as well as last year, you have your general data quality assessment and the data pre-processing. You filter the sequence data and perform genome assembly. There's a really great workshop from last year's MMID coding workshop that shows genome assembly if you're interested. And then taxonomic classification, which was part of our March 30th workshop this year. And then your sequencing data is ready for downstream analysis. So different things like characterization, looking for antimicrobial resistance genes, virulence genes, looking at the phylogeny of the sequencing data, which is what we're doing today, as well as visualization, which we're going to focus on phylogenetic visualization in next week's workshop. So this is just an example of phylogeny that I made. Phylogenetic trees are used to describe the relationships among groups of organisms, which can be constructed based on the genetic sequences of the organisms. A rooted phylogenetic tree presents a mode of evolutionary history that's depicted by ancestor-descendant relationships, between the tree nodes, clustering of sister or cousin organisms at different levels of relatedness, kind of as shown as the figure here. In infectious disease research, phylogenetic trees are usually built from the pathogen's gene or genome sequences in order to show which pathogens sample are genetically closer to another sample. And it also provides insights into the underlying observed epidemiological linkages and potential sources of an outbreak. There are two different, I guess, main types of methods. There is the character-based method, which is a mathematical model that describes the evolution between genetic characters. And this character-based method compares all sequences simultaneously, considering one character or nucleotide site at a time. Examples of these would be maximum parsimony and maximum likelihood trees. These methods use probability and consider the variation between sequences. There is also distance-based methods. So these are based on the matrix of pairwise genetics distances shown here, and they're calculated between sequences. So this would include the unweighted pair group method with arithmetic means and neighbor joining trees. There's many different types of tree tools out there, probably hundreds. Today, we're going to be using IQ-Tree. It's very efficient phylogenetic software that builds maximum likelihood trees. Another tree building software I've used in the past is MashTree. While this doesn't infer phylogeny, it creates really fast comparison between whole genome sequences, and you don't need to create a multiple sequence alignment first. You can run it just off of your FASTA or FASTQs if you kind of just want to get a snapshot of the orientation. There's PhyML, which is also a maximum likelihood tree method. There's QuickTree. These other tree building methods I haven't tried out yet. There's Beast, which is also very popular. It's based off of Bayesian inference, relaxed molecular clock, and demographic history. There's Clustal W, and as well as mega. So you might have heard of some of these before. Okay, and now we're going to go back to our terminal. Okay, so now that the file should be unzipped, we're going to install IQtree. So whatever environment and directory that you're in. Second. For me, because I am currently working on waffles, I'm going to use Slurm work flow manager. So you can go to install. If you are not working in waffles, you can do conda install -c bio conda IQtree. But because I'm working in waffles, I'm going to add sbatch -c, use one core. Have not yet memorized this string. And then we're going to put it in quotes and press enter. Okay, so that should be running. You can also check if the package was installed by looking at your conda list and press enter. So it looks like it's still running, so I'll just give that a few minutes. Does anyone have any questions? Okay. Yeah, for anyone not working in waffles, you don't need to include the sbatch. So you would just put conda install -c bioconda IQtree. Looks like it's still running. Looks like I did not change into my phyllo directory. So I'm going to go into there. While that's running, and I'm going to make a new directory inside phyllo, which is currently empty, called MSA. So make directory MSA, and it's right there. And then I'm going to go back to where MSA file is, which is in my workshop folder. And I'm going to move it into the MSA folder. I'm going to do move MSA.FASTA. We don't want the zipped one. And I'm going to move it into phyllo MSA. Then I'm going to go into the MSA directory and check that it's there, which it is. So that's great. Let's see if the package is done. Yeah. So when you do conda list, you can see that IQtree is downloaded, and then as well as any other associated packages that it downloads and that it needs. So I'm going to go. I'm going to pause there and go back to slides. I think I jumped ahead a bit. So you should have the IQ tree hopefully downloading or downloaded already. So what is a multiple sequence alignment? So that's what we're going to be working with today. The multiple sequence alignment is basically an alignment of all the biological sequences. Today, I think we're working with 15 different sequences of similar length. And then they're used to infer homology and evolutionary relationships between sequences. So IQ tree and a lot of other tree programs, they take a multiple sequence alignment as the input, and then it's able to construct an evolutionary tree that is best explained by the data. So there's different formats for multiple sequence alignment. Ours is in FASTA format, so it'll look something like that. But there's tons of different types of formats that you could create. Since we didn't create the multiple sequence alignment together, there are tons of different tools that are out there to create multiple sequence alignments. Again, there's Clustal Omega, MAFT, K-Align. I used the Genome Taxonomy Database Toolkit, which I actually really like, but this tool takes about an hour just for the 15 that I ran. It's actually a taxonomic classification tool. So some of the workflows that it runs with, it'll align the sequences. That's where I get the multiple sequence alignments. It'll identify them. So it'll give you like, it'll say Vibrio parahemolyticus down all the way down to the species level. So it's tons of different things in one, but we just didn't have time to do that. So that's why I like using that tool, because it gives me lots of different information. So the data set, the sequences that I pulled are from the same article that Jill was working with the last two workshops, with a focus on this cluster in red. And then I pulled a couple other sequences randomly from the tree so that we'd have a cooler looking tree once we get to the visualization stage next time. Okay, we'll go back to the demonstration. Okay, so we should have our MSA folder. We moved the FASTA file into our MSA. And now if we should be able to activate the, I previously should activate whatever environment that your is built in. I had previously called it IQ tree, but I'll do that. So return to the phyllo directory. And we're going to make a new directory called IQ tree. So make directory IQ tree, and that's where we'll move our output to after. Go. And then we're going to go into IQ tree. It's currently empty. And then we're going to be able to run the IQ tree tool. So if you're not working in Waffles, you can type IQ tree -s, which will point to the multiple sequence alignment we're looking for. So the multiple sequence alignment should be back a directory. It's in MSA. And then MSA.FASTA. And then you can press enter. For me, since I'm working in Waffles, I'm going to sbatch that. Type. Oops. And press enter. So now that should be running. Oops. Okay. So there's different output that IQ tree produces. There's an MSA, or there's a .IQ tree extension. And so this is the main report file. It'll show computational results and contextual representation of the final tree. The .tree file is what we're most interested in. This is the maximum likelihood tree in Newick format, which can be visualized by any supported tree viewer program, which is what IQ tree states, which is really important. And then there's the log file, which you can refer to. It has the log of the entire run, and you can look for errors if anything goes wrong. It's good to go back and look there. Okay, go back. Might be done. Press. So if you press LS, you probably are not going to have the output in your directory. That's because it might have left the output in the MSA directory where the MSA file is. So let's move to the MSA directory and check if it's finished and it's in there. So it looks like we've got a bunch of new files in there. So I think it's done. So we're going to move just the tree file that we're interested in to the IQ directory, IQ tree directory. So to do that, we'll type move, we'll star just the tree, tree file. And then we're going to move it to IQ tree, just so we have it kind of all by itself. And then let's go back to IQ tree. So now we have the msa.pasta.tree file. So now we're going to open up our browser. And we're going to go to this website. If you type in Fandango, it should be the first thing that comes up on Google, or the link is there. Go. Okay. So for me, this is what the website looks like. It's an interactive visualization of genome phylogenies. It's a very quick way to kind of view your tree output when you're done running a tree. And so once you have this open, go to your directory that has the tree file in it and grab it to go to my workshop, Philo, IQ tree, grab the tree and just drag it into Fandango. So let me know if you guys have an error when you do that. Everyone has an error? Okay. So interestingly, we made our tree. It's in Newick format, and Fandango is supposed to be able to take Newick formats, or as we saw, IQ tree is supposed to create Newick format trees that work in many different tree visualization tools. If you kind of go and read in the docs, it actually states that Fandango wants the extension to be .tree or .tre, which is kind of interesting. I've also found that .Newick or .nwk works well. And sometimes just changing the extension is all you need and it'll work. So let's go back to our code. And let's change the extension. So to change the extension, we're actually just going to make a copy of the file. So we'll go CP for copy, type the MSA FASTA tree file, and our new name is going to be MSA.FASTA, and we're going to call it .tree instead of tree file, and press enter. So now we're going to have a copy of the file. We still have the original there. And we're going to go back and we're going to try and drag that into Fandango and see what happens. Okay. And it's also going to give you a heads up that the phylogeny was loaded, but without any recombination, pangenome, or metadata files. That's okay. So we'll just kind of click out of that. And now we see kind of like a little tree based off of our sequences. Is everyone able to do that? Awesome. So if we look at the tree that we created compared to the original article, obviously it's not the same because we didn't use as many sequences as the author, just so they would go a bit faster. But we do see that main cluster, and then we do see a few other branches. So that's great. But what we're still missing is the sequence metadata. Metadata is super important. It includes the information about each sample that's at each tip of the tree. It basically exists to give the data context. So it's very crucial to understand how the sequences are distributed across environments and the processes underlying the observed patterns. So in this article, the author actually did summarize the metadata in table number 1, which is really great because it's important for data reusability and reproducibility. And we'll be able to make use of this metadata in next week's workshop. So I'll compile it into either a TXT or some sort of Excel file. So we'll be able to use it next week. But we're not going to use it today. Some of the information they included were the strain, servar, MLST type, location, year, sample, and this presence or absence of gene in the last column, which is TDH and TRH, which it's not really important for the workshop, but it's just good to know if you were the author looking at this. Adding metadata in these kind of interactive type web programs like Fandango is a little bit limiting. They usually require you to have a very specific file format, as we already saw for the Newick tree, which kind of limits the type of visualizations you can do. Usually, it's very often limited to bar plots or pie charts, as well as it's just less customizable overall. And some of the programs may require you to pay or have a subscription in order to save any of your annotations, which is just, not realistic. So some examples of other tree viewing tools are iToll. I do like this one, but you need to pay, I think, to save some of your annotations. But if you make an account, you can save a lot of your basic just trees in there. Osspice is very good. Fig Tree I haven't used, but it's very popular. And then GG Tree is what we're going to be using for next week. And that's like fully customizable. And since this is a coding workshop, there's coding involved, and we'll get to explore that a bit next week. And that's actually it. So we're done actually pretty early. Some helpful resources. If you want to read more about building a phylogenetic tree, about the different algorithms and applications, and how trees are constructed, I left some resources here. And there's also a lot of really great resources throughout the slides. But also just kind of a heads up for next week. So we're going to be working in our studio. And we're going to need the ggtree package. So from my experience, sometimes it can be a little tricky downloading or installing ggtree. So if you're planning to attend next week's workshop, it would be great if you could try and install this ahead of time. It does require, it's installed through BioC Manager. So if you just type this code here, BioC Manager, I don't know what that's called, install and ggtree, you should be able to grab it that way. There's also instructions on their GitHub, as well as on the link provided here. That way, if there's any sort of troubleshooting, maybe we can, you can email us and we can try and figure out how to troubleshoot that ahead of the workshop. But any other packages that we need should be fairly easy to install. But yeah, thank you everybody for attending. It's just a quick workshop today. Make sure that you fill out the exit survey. The link is there, and we would really, really appreciate any feedback you have for us.